Hello, everybody. Good evening. Can you confirm if you can hear me all right? Okay, perfect. Great. Thank you. Alrighty. Um, let me just share my screen and just confirm if you can see my screen also, and then we'll start talking a little bit about what this session is for. Okay. Right, everybody. So, good evening. And so you've all signed up for this course, Cell Culture Technologies, right? Um, as you may be familiar already. So this is an 8 week course, and you'll have 5 lectures of roughly 30 minutes each. So that's about 2.5 hours every week. Now these lectures are released every Friday, and then I think about like 12 days later on a Wednesday, there's an assignment which is due for that week. And so like the week one lecture were released, I think, last Friday, so about 10, 12 days ago. And then today we have this live session. The idea for these live sessions is that you already have had those live sessions. You've seen those, oh, sorry, the recorded lectures, sorry. And then this live session, once you've seen those videos, if you have any questions, doubts, you can ask those. Also, I would anyway be going through the general topics, general concepts that were talked about in that week. And then we will see some questions which are similar to what you would see in your assignments. So these questions would be not exactly like the assignment, but you'll get an idea of how to solve your assignment questions. And then starting next week, we'll also take a look at the previous week's assignment solutions, and if there's any question, doubts, something incorrect, then we can just work on those. All right. So so before we start the actual things, would some of you like to tell me if you've had attended NPTEL sessions, or if you've attended these live sessions before, and if there's anything in particular you would want me to take care of that you've not had a good experience with earlier, or if you've had some good experience and you just want that to continue here. Also, I would like to know, ideally everybody, but if some of you can tell me what is your background in, so that do we need to go into some more basics of this course that were maybe not covered in the recorded sessions, or are you already familiar with a lot of basics and you would like to skip over something and discuss something else? So either way, any suggestions that you have, please feel free to unmute yourselves and talk, or you can also put your suggestions, questions in the chat box. These sessions are, by the way, they're being recorded. So what we'll do is, you have these live sessions. Okay, by the way, before I go, so there are going to be two live sessions, one on Mondays and one on Tuesdays. So I'll be taking the one on Mondays. The link stays the same, the Zoom link, and there's going to be another one on Tuesday. So that's starting tomorrow. I think that's on Google Meet. Both of these sessions would be complementary. So like you don't need to attend both of them. I mean, it's up to you. Um, and the attendance in these sessions is also not necessary. These are just an additional thing to help you out. So these sessions are being recorded, and I'll be putting the recording on the YouTube channel right after the class, and I'll share these links with you. You'll also have these links on the course page, so don't worry. And any material we discuss, so for example, this PowerPoint that we have today, all of that I'll put on a Google Drive, and you'll have the link for that as well. So yes, so you do have, in addition, so maybe we can talk about some books that can be there that are on this topic. But other than that, the final exam mostly looks very similar to what your assignments have been like. So every week you'll have your actual assignment, and you'll also have some questions that we will discuss in these live sessions, both Monday and Tuesday. I think the questions we'll talk in Monday and Tuesday would roughly be the same. But either way, these practice questions and the assignment questions and your actual assignment notes, the lecture, the video, whatever you see, I think those would mostly suffice. But of course, we can talk about some books that can be good. So again, for these problem solving live sessions, it will be good if you've already seen those lectures so that we can build on those and not discuss the absolute everything that was talked about in these lectures. All right. So again, I'll give you a couple of minutes if any of you want to talk about anything. Suggest, tell me about your background, or if there's anything in particular that you're looking out of these, or this course in general, or these live sessions. Yeah, let me know. Again, unmute yourselves or put it in the chat box. Anything works. Because these sessions are meant to be interactive. So it's nice if you can do that. Otherwise, no problem. I'll just speak to my screen. Yes, I think so, Shreya. Yes, the exam questions, as far as I've seen for all the previous courses that I've been a sort of like a DA for, the questions are very similar to the assignment. And okay, you're a B biotechnology graduate. Good. Oh, by the way, I am Pallavi Gupta. I am a final year PhD student at IIT Delhi. So my PhD work is using stem cells. We can talk about more. I would love to talk about some of my work once you're more familiar with the actual cell culture technologies. But yeah, my work is in cell culture technology as well as a little bit of bioinformatics to analyze some RNA sequencing data. So I'm a TA for this course for the first time. Previously, I've taken some bioinformatics courses, mostly that were based on R. I'm not sure if any of you have been in those courses, but yeah. So yes, already a disclaimer. There could be places where I may be wrong. So stay attentive and let me know if I'm making some mistake. All right. And yeah, all right. So if there's one final time, if there's anything else, and again, please feel free to unmute yourselves at any time during the discussion. We have been allotted like a 2-hour slot for these live sessions. So I will be here for at least 1.5 hours, if not more. Um, for the first class today, we might not have a lot to discuss, but anyway, I'll still be here. So whether right now or towards the end, if you have any questions, can pop in at that time. Um, yes. So for the recordings, yes. So these sessions are being recorded. And okay, let me go through something else before we start. So this is your course announcement page, right? So last to last week, your week one recordings were released. And this is where everything is. This is where your assignment is. So this assignment is due in 2 days. So whenever you have these sessions on Monday, Tuesday, the Wednesday after that is when your assignment is due. So that is why these live sessions are arranged. Like they're arranged such that you have watched the videos. You may have some questions when you're trying to attempt the assignment. So now you can attend these live sessions, ask your questions. We will obviously not be discussing the exact answer for these directly. So, but we can talk about why would something make sense if we're confused about some question. Uh, do the assignment, and these assignments are especially important if you're planning on getting a certificate for this course. Right? I think one of the deadlines for the certificate was today, and the last day with the late fee is I think the 29th of August, if I've read the announcements correctly. Either way, assignments are good so that you know that what you've learned in the sessions, are they making sense? Were you able to grasp the concepts and so on? So yes, that's the assignment. That's one deadline you should remember. The other thing is here there is a Q&A page. So if you click on this, it's going to redirect you to a Google group. I think somewhere here is going to ask you to join the Google group so that you get emails every time somebody is posting a question or somebody is giving an answer. If you do have any questions apart from these sessions, like if we were not able to talk about something or if there's something, so I'm not setting up the assignments. So particularly if there are any questions about the assignment or about the course otherwise or the videos directly, this would be the best place for you to ask those. And you can see here for some of them, people have already gotten answers. So yes, that's the Q&A or the forum. Definitely join that and actively use it. The other thing is, so here below you see a problem solving session link. It says the recordings here, and this is going to redirect you to this Excel sheet. Right now, this is empty because this is the first session we are having. After today's session, I'm going to put the YouTube link for this week's lecture. I'm going to put the Google Drive link here. So we are two TAs for this, one me and the other person's for tomorrow. We'll tell the attendance that okay, there are about 27 people who've joined today. So uh, that's what, so here you'll get the link for your recordings, right? So yes, so again, the attendance is not necessary for your assignment because we understand you might have something else, some other tuitions or classes and all. And these courses will definitely help you get an idea of the concept, right? So maybe, of course, there could be questions that people ask in job interviews that you might get help from in these courses. So directly, no, but yes. But and of course, in your CV, you can write that you've attended the lectures for this course, right? So it helps in your CV, and I think these are conceptual courses. So we won't be really focusing on rote learning. I don't want like me or your course instructors, the whoever like Professor Mehnat does. I don't think he would be interested in you to be rote learning that okay, these are the 5 types of things you should remember, right? And so yes, so the recorded lectures, so there are 40 lectures in all for this course. So every week, every Friday, you will have one set of 5 released. So these 5 were released last week. And then week 2, these were released 3 days ago. The coming Friday, week 3 lectures will be released and so on. So you watch these lectures, these are about 30 minutes long each. Watch them. You can, I mean, I would suggest you to directly attempt these assignments. And if you get stuck somewhere, then you have these live problem solving sessions where we will discuss the concepts. So if you look at the outline for today's session, we'll have a general discussion about some topics. It's not a lot, but a few. And then we have some practice questions. And then starting next week onwards, once we have the assignment 1 solutions released. So assignment deadline is on a Wednesday, and I think by Thursday you will have your assignment solutions released. Take a look at those. And then if you have some questions, then the next session, we will also go through those and see if all of them are making sense, right? And then we just end. So again, 1.5 to 2 hours is what we are targeting. Feel free to ask questions again at any time. These are meant to be interactive sessions. So if you're able to unmute your mics, well and good. Otherwise, like you've been chatting on in the chat box, it's perfect as well. All right. Any more questions? So yeah, the Zoom link for Monday session stay the same. There's another link for the Tuesday Google Meet that also stays the same throughout. You can see that the session is being recorded. So I'll put this on my YouTube channel. The one on Tuesdays will be put on another YouTube channel. But again, there on the course page, you'll have the link to everything. All right. Yes, I am recording this. Yes, so this will be available. So your recorded lectures every week, they're already there. And the Zoom meeting and the Google Meets, again, you don't need to attend both of them. Monday, Tuesday, whatever is available for you, you can attend that. And you'll have all these links. Let me anyway share these links with you in the chat box today. But you can anyway refer to those through the course page. In terms of whether the course is recognized internationally, I am not very sure, but this would anyway be a part of your CV. If you're talking about the certificates being like, I am not sure if can be used like for a credit transfer or something, but you have anyway attended this. So it adds to your list of, the work you've done, the things you've read. It's as much as your transcript. But I am not 100% sure if these are recognized internationally. But you will anyway benefit from them, uh, whether or not you choose to take the certificate. Like NPTEL courses are mostly designed in a way that these would be in addition to um, your usual curriculum, because these are smaller courses. You just have like some 4-week, 8-week, or 12-week courses. So they're a little quicker, faster. Um, but yes, again, the direct answer to your question is I'm not sure. I will try to see if I can find some information on this, but as of now, I don't have the answer. All right. Okay. All right. Great. So a lot of you have been active so far. I would request you to continue that throughout this session and the upcoming ones. Okay. So the course give me a second. I think the exam fee, there's one date of 25th of August, which is today. And I think including the late fee, it is the 29th of August. So if you look at the course page, in the announcement page, there are already all these dates mentioned. So definitely go through those. And to get the certificate at the end, I think it's important that you have scored at least 80% or something out of all the assignments. So take care of that. Do not miss your assignments. All right. But again, the exact details of the specifications of the NPTEL structure is something you'll have to check. You can always email the NPTEL people. There will be some contact page available. That should help. But I think there's one last date, which is the 29th of August. That's like the final last date. All right. Okay. So in the first lecture for this course, um, so sir started off with talking about what is like why would you have a theoretical course for cell culture technology? Because so okay, before we go there, so cell culture technology, in simple terms, is your body is made up of cells. Okay? Different types of cells. We'll go through that sooner in different classes. So if you want to, if you want to study something, okay, you can study it just by looking. Okay, I have some skin, I have hair, I have a brain, heart, and but you also want to look at something under the microscope. So you can take out a piece, dissect, look at it under the microscope. Okay, but then once you take it out, it's going to be dead. Okay, because within your body, it's getting some things which is keeping it alive. Somewhere in the course, it was mentioned that some cells live for some time. Okay, maybe 10 days, maybe 30 days, maybe 6 months, maybe longer than that. So whatever it is, they're alive for some time. They're doing some function. Now, what are those functions? What is keeping them alive? What is happening if they are dying, when they are dying? What all are they doing when they are alive? Is a cell existing by itself? Is it making some contacts with something else? And so on. Everything, a lot of it might seem very obvious now. Okay, for those of you who've had some biology in school, like biology courses in your school or in your undergrad or more, a lot of this would seem intuitive. Yeah, okay, it's very simple, it's very intuitive, it's very basic. But a lot of this came from some very simple studies where you're simply growing cells outside where they are normally existing. Okay, and when you say growing, it means they're alive. Most likely, they're also dividing because you start off with the cell. If they are okay, there are two options. One, you have a master cell and they are just staying alive. You start off with 100 cells, you stay at 100 cells. Or you start with 100 cells, maybe you get more, maybe you get less, depending on whether the rate at which they are dividing is more or less than the rate at which they are dying. Okay. They could only be dividing, maybe none of them are dying, maybe more are dying than they are dividing, or maybe there's like this homeostasis, there's equal, the rate of division and the rate of dying is similar. Okay, anyway, and please again, if you think I'm throwing any jargons, please feel free to interrupt, ask, or you can put it in the chat box. For those of you who have joined us recently, this session is being recorded. I'll just quickly go over because I can see a lot of people have joined the last 5 minutes. So every week, five recorded sessions are released. We'll have a doubt clearing session on Mondays and Tuesdays. I'll be taking the one on Mondays, 6 o'clock, Zoom link. These sessions are being recorded, and any material that we discuss here will be shared on the Google Drive. You will have the link to all of this on your course page, so don't worry. So we've just started the actual discussion for this week's live session right now, right? So you're culturing cells, you're growing cells, we'll just call it culturing, outside where they exist. So in this course over the next 8 weeks, what you are going to be learning is about some concepts and theories, some basics of what is used, what do we know, okay? Of course, how things were built, but also what currently is being used in this field. Okay, because it doesn't make sense that you're starting from scratch every time, because people have already published protocols and papers and findings. So we can already build on this. So we will talk about what all is known. We'll talk about what is still unknown. We'll talk about where we have, okay, we know something, we are building on it, but we know for a fact that this is not real, but we still don't have a better choice. Like we still don't have a better alternative. So this is what we have chosen. Okay. So why would you want to culture cells, which is just what we saw? So you want to learn about what the cell looks like. Okay. So can anybody tell me when I say a cell, what sort of scale or dimension am I talking about? Is it kilometers or millimeters or micrometers or nanometers, femtometers or decameters? So what scale are we talking about when we talk about a cell? Any idea? So micrometer. Is it 1 micrometer? Is it 100 micrometer? Could it be both? Could it be less than a micrometer? Could it be more than 100 micrometer? So you're all correct. It is in the micrometer range. And yeah, so you do have small cells, which can be as small as 1 or 2 micrometers. Yes. So typically, yes, cells are, when we talk about, so again, it was very much emphasized during the lecture that we'll only be talking about animal cells. Mammalian, so human, mammalian, as well as amphibian and all. So when we talk about that, we are mostly talking about more than 10 micrometers or 10 microns and something around 100. But that's the range we'll be talking about. So of course, so that is something that you can't look directly through your eyes. So you need a microscope for that. So you want to study that under a microscope. There are going to be lots of things you'll have to do, because even if you can see a cell, there are structures within a cell which are much smaller, which can be in the nanometer range. Yeah. How do you look at that? Okay, you can say, okay, I will use a better microscope. Cool, you can do that. But then how do you distinguish? There's going to be maybe just like 10 different gray lines, 10 different gray structures. How do you identify what each of the structures is? What is each of the structure doing? Right? So just using a microscope is not enough. You may want to stain some of those things, okay? Or you may want to have some sort of a functional readout. You're not just looking, but you're getting some data that, okay, I gave it compound A and I got compound B. Okay, so there's some reaction happening. So for that, you don't necessarily need a microscope. Okay, again, we'll talk about those. I'm just throwing some sentences for the time being. And again, let me know if any of those sentences don't make sense. So you want to talk about the cell structure. You also want to look at the growth. And we'll see why looking at the growth is important, because some cells don't divide. And then if there is an injury, so if you get injured on your hand, if you get a cut on your hand, it heals, right? Because when you got a cut, some cells died, okay? Or when you get a biopsy, something gets removed, you get an infection. Suppose something's happening here and you have had to remove a part of your skin. It heals over time. It may take a month, but it will heal because some cells were dividing. But people talk about, okay, injuries to the heart or injuries to the brain are tough to deal with because those cells don't divide, right? So there's nothing healing them back. And then those structures are also very complicated. Skin is relatively less complicated. Fewer number of things, so as to say, not exactly correct. And then more complicated structures. So even if things were dividing, you want them to be exactly how they were earlier. Again, we'll talk about these things in more detail in the upcoming classes. But then growth, that is why growth is important. How do you ask something to grow when it has lost its ability to grow? Okay. And then, of course, that everything, most of biology, somewhere or the other, has to lead to talking about diseases, treating diseases, preventing diseases. Okay. So, broadly, everywhere, this can be an application. Also, so again, we're not going into details right now, but when you talk about antibodies, so you've all been vaccinated against something or the other. Okay. Um, those vaccines, those things, a lot of them may have been generated using some cells that people are growing in the lab. Okay. So you have cells and you've tasked those cells that, okay, so in your body too, you get immunity, right? So taking the case of COVID-19, even if there was no vaccine, some of the people, they got immunized against COVID-19 because once they got the virus, their body recognized the virus, it made antibodies, and now the next time that the virus enters, the body is already prepared because it can identify the virus as a foreign object. So that's what vaccines also do. You don't want every disease to be like COVID-19, where you get one infection and then your body is prepared. You want that, okay, instead of getting the actual infection, you get something which looks like, molecularly, it looks like that infection, but it can't cause you infection. So you give that to the body, that's the vaccine. The body says, okay, I know this is a foreign object, I'll deal with it whenever need be. And then, yes, if at all you get infected, then the body knows, okay, body will be ready very quickly. So now those things need to be created at a large scale. So there are cells which in your body can do that, and then people have trained the cells to actively do that outside the body also. So in these cases, now the cells, normally if the cells are doing 10 things, one of them being making antibodies, in the lab, you're like, okay, I don't need the cells to be doing all 10 things. I just want the cells to be working on doing that one task of generating the antibody. And I don't care if the cell does the rest of the 9 things. So the cell here may not be identical to the cell that's there in your body, but you don't really care as long as it's doing that one task correctly. Just keep this example in mind. Sensor, again, cells are able to, we'll talk about this in more details again, but cells are able to detect things in their surroundings, whether that thing is coming from the neighboring cell, whether that thing is coming from just the surrounding. So cells can be used, cells can be used as sensors where they detect something, and then you just want to read out. You want, okay, if the cells are making some compound, they'll say, okay, that means it detected something. Or if the cells are dying, then okay, that's also some sort of a sensor because it's able to sense and then it's dying. Give me a second. So yeah, so cells can be used as a sensor. Two terms, personalized medicine and regenerative medicine. I think personalized was not actively talked about in the lecture, but we'll just come to it. So personalized medicine means, I'll give you a very brief example. So there are medicines available for a lot of diseases. Okay? But then, do you know how these medicines or how these treatments work? A lot of them happens through clinical trials. Okay. Now, for clinical trials, you need a lot of people who have that disease so that you can check the efficacy of that drug or that medicine on them. Now, what if there is a disease, these diseases are called rare diseases, which just happen, we know just 10 cases across the world for it. Oops, sorry. Yeah. So what about there's some cases where we know just 10 people who suffer from a particular disease. So in that case, it can be tough to develop a medicine just for people, just for 10 people. And this is what happens. So big medicine companies would not want to invest when their market is just 10 people. If the market is 1 million people, then yes, it makes sense to invest money. That's one case. Another case can be maybe there's a disease which affects many people, but we still haven't found drugs which can treat everybody with those diseases because there are just so many multiple subtypes. Some drug works on somebody, some another drug works on another person, and it's like a hit and trial. For a lot of, I think, neurodevelopmental diseases. So let's talk about something like epilepsy. If you look into it, there's a lot of subtypes. Okay, and then there are a lot of medicines available, treatment plans available. But then what happens is either the person's not responsive to it, or it may happen that they develop resistance again. So initially the medicine was working, but now it's no longer working. So what the general plan is, okay, give one medicine, observe for some time, maybe a week, maybe a month. If it's working, great. If it's not working, you switch to medicine number two, and then the cycle repeats. Okay. So what happens in that case is that you just lost, you don't know, you're just, it's a hit and trial on the actual person, on the actual patient. So personalized medicine is the idea that you take cells of that patient, you grow those cells in the lab. So for the brain, you're able to grow brain cells. We're not discussing how you obtain the brain cells right now, but you're growing the cells of the person, of the patient in the lab. And then you can test. So you can grow tons of those cells in the lab. Okay, just give them the right whatever things, and the cells are going to grow. And then you can check 10 medicines at the same time. You have 10 batches of cells, you have them in 10 different compartments, you give each of them one drug, observe, see, get some readout, and whatever works, you can offer that to the patient. So at least you've narrowed it down that, okay, again, there are lots of things. Maybe the cells are not behaving exactly the patient's cells would, like the patient's brain would. But it has to start somewhere. So things like this are what personalized medicine is about. Regenerative medicine, on the other hand, is so like we talked about injuries to the heart or the brain, they cannot be treated on their own because of something. The cells don't there, they don't know how to divide anymore. Okay, they're like, we made the final thing and that's it. That's how it is. Okay, evolutionarily, that's how it has been. Now, regenerative medicine is in the lab, you can make those types of cells which know how to divide, and you give those cells back to those organs of the patient, and they have the tendency of those cells is to divide, and again lots of things. But if those cells are able to divide into what that organ needs now to recover, then your task is done. Okay, so so the first three things are more intuitive. The last two, they are relatively new things. They both require something known as stem cells. So stem cells are those cells which can make anything. So again, going back to how does life begin? So you have a zygote, which is just one single cell made of the egg and the sperm. So that single cell gives rise to everything that you have. Okay? That divides, that's one, it becomes two, it becomes 4, 8, 16, and so on. Now, initially, all of that single cell, also those 16 cells, all of those can become anything. Once there is a big mass, once they have, I don't know the exact number, but let's assume once they are at something around 10,000, is then when they are divided into some lineages. So there, if you've heard, there are lineages like endoderm, mesoderm, ectoderm. Then you have blood lineage, brain, and heart, and then within the blood also you have 2, 3 different lineages and so on. So then each way you have like an inverted tree, okay, where you have more and more uh, branches or more and more roots that are coming up. Okay, so far, do you have any questions? All good? Okay, I'll assume everything's all right. Okay, so so the so one of the things that were talked about in these lectures were whether cells come from pre-existing cells or whether just cells emerge. So it was not discussed in much detail also, but then yeah, so cells come from pre-existing cells. If you have one cell, then it divides to something called mitosis, uh, and it becomes two cells. Now the two cells, they may have the exact same identity as the previous cell, or like initially, yes, initially both the cells will probably be exactly the same as that parent cell. So parent and progeny, or parent and daughter cell, or mother cell and daughter cell. These are the terminologies you might hear. So initially, those two daughter cells, they will resemble the parent cell. But then whether they continue to remain that or not depends again. So when we talk about that, okay, these cells, they're being divided into different lineages or different identities. Oh, yes. Hi. So the session's being recorded, and after this session, I will upload the recording on YouTube, and you will have the link to that YouTube recording in your course page. So if you scroll down, there's something known as a problem solving session there. So you can get the recording there. All right. Okay. So one of the first, so that was the idea that cells come from pre-existing cells and they're not spontaneously emerging. So again, one cell can become two cells. Whether those two cells resemble the parent or not, it depends, or whether they continue to resemble the parent or not. When the cells are being divided into lineages, very often it happens that out of the two cells, one will remain identical to the parent, and the other one will become what we talk about, what the other one becomes differentiated. All right. So that's yeah, so that's one thing. So one of the scientists, Dr. Harrison, he established one of the first, so this is considered to be one of the earliest cell culture experiments. So what he was doing is he was looking at nerve tissue of the frog. And now cells need something to support them. Okay. And by support, okay, two things. Cells need something to grow in. Okay? They need food. Normally, that food is provided by blood. Okay, in the body, um, it's provided by blood. Nutrients, oxygen, both the things is given by blood. And if cells need some signals to decide what they want to be, then those may be provided by their cells which are next to it, or it may happen within, the parent cell, daughter cell, things like that. We'll go into more details, but broadly, cells need a lot of things to survive. So the neural tube, again, I don't go into the details of what a neural tube or what neural tissue culture they did. It's a very nice story, so you can read about it for sure. But for the purpose of this course, we are not going into much detail. So this was cultured in lymph medium. So the lymph medium that's there, it also has a lot of nutrients. So the neural tube was grown in that medium. And then some cells need some support to grow on. They just cannot grow floating. Okay, we'll talk about that also because it was discussed in the lectures about adherent and non-adherent cells. So they need some sort of a support. So in case of this nerve tissue, spider web was used as a support. So right now what we're talking about is not microscopic. The I think this was tissue culture. So it's okay. So yeah. So another thing that was talked about was tissue culture and cell culture. So cell, when you're talking about when you have single cells to begin with, is generally when you say cell culture. And when you're starting off with a chunk of cells altogether and you'll just let them be as chunks, then it's mostly tissue culture. But they're very related terms. A lot of the concepts, majority of the concepts that are applicable to one will be applicable to the other. So we'll not just like demarcate those two things a lot. But yes, just to know if you're dealing with one or more cells, also when you're dealing with cells which are just dissociated, when it's a cell, the cells are not in contact all the time, then you're talking about cell culture, and then you're dealing with, you're never dissociating the cells, always have them together, that's tissue culture. So yeah, so that tissue, so something which is visible, not microscopic, that was supported by, again, spider web, which is also very much visible. But then now what you do today is you have cells which you can't see, you grow them on something, on a supporting something, which is also something you can't see. That what exactly is it that you're, what exactly is the constituent is also something you can't see. Okay. So this is what the example you was used in the lecture, that you have an animal, you have an organism, you dissect a mass of the tissue, and you can directly grow that intact piece. So that's going to be called tissue culture. Or if you dissociate them into single cells and then grow them, that's going to be cell culture. Now, when you talk about a tissue, for example, take your skin tissue. You take a part of the, if there's a biopsy for whatever reason, and if you look at the cells under a microscope, whether you stain for something or not, there's different types of cells inside it. Okay. Let's say there are 5 types of cells in your skin. Okay. Some of them, some of that is your hair, okay, the hair follicle, all those things. Some of them is, you have glands, right? You have sweat glands. So there are cells which are making up those glands, and they're going to be secreting something out. There are going to be cells which give the color. So melanocytes, that was also talked about. These are the cells that have that that are the pigmented cells. Then, of course, you have cells and multiple cells which are giving the support. Okay. There are also nerves, because you can feel the touch, right? You can sense hot, cold, prick. Okay. So there are lots of different sensing cells also. But let's say there are 5 different types of cells. Okay. 5 different normal functions that they do. Now, do you think, again, this is a question, so I would like one or more of you to answer this. Do you think all of them have the same requirements, whatever requirements, to grow and be what they are? Are they getting the same type of signals? Do they need the same kind of food? Do they give out the same kind of signals? And so on. Do they want support? Do they not want support? Do they want the same kind of support? Most important is, do they want like the support and the food and the signal? These 3 things. Do you think all of them have the same type? Yes and no. And would you like to say something about why you choose yes or no? It's an open-ended question, so it's up to you. If if you would like to just say out what you think. Anybody? No, nobody. Give it a go. Otherwise, these sessions are going to become boring. I'm just going to be speaking. So I need to get some inputs to be able to speak something else. Otherwise, I'll just keep on speaking what I have in mind, and then maybe we miss out on some good discussions. It's okay if what you say is wrong. It's just 30 people watching. No problem. And it's okay if you're wrong. No problem about that. The question is, if your skin has 5 different types of cells, um, do they all have the same requirements in terms of the signals they want to be what they are, the food that they want, and the kind of support, literal support uh, that they want to be growing? Come on, anybody? Yeah, no, okay. So they're not identical, right? Would you or would anybody want to elaborate on why you think so? Well, so they are different types, right? It's sort
Excuse
of,
me, ma'am.
yes, yes.
Yeah, ma'am. I had answered that they would not be identical, and answered as no. And I think that if we have different types of cells on a face, I mean, they would. I mean, I'm talking in the perspective of like how they like mature and differentiate. If I believe that we have different types of cells, and they would differentiate in and perform a specific function, so they would have the requirements which will help perform that specific function
Right.
efficiently. So
Right.
that is why I think they have different nutritional requirements as well as signaling support.
Good,
Thank you,
good,
ma'am.
good. Perfect. Perfect. That's a very good answer. So yes. So so the story begins when they were all the same kind of cell, when they were a stem cell. And then stem cells divided, and some of them became type 1, 2, 3, 4, 5 different types of cells. So they all got different signals, and that is why they became a particular type. And they remain a particular type because probably they are getting the same kind of continued signal. Also, whether or not they're getting the continued signal, once they've become a particular type, they do some task, okay, whatever their function is, secretion, growth, releasing pigments, whatever. Okay. So all of them have these specialized tasks. And as you said, uh, they need different things that would enable them to continue doing that task. Ultimately, a cell is a structure which is performing a task, essentially, right? It's a machine, a biological machine, right? I think that's how cells are also often described. So, so yes, so these five different types of cells, they need different things. Now, if I take that uh, chunk of cell, a chunk of the tissue, and if I want to grow them, grow that chunk, grow the cells from that chunk, whether or not I dissociate them and then grow them, or whether I just put that intact piece together. Oh, okay. By the way, where am I growing these cells in the lab? So I don't have a figure right now, but what we can just do quickly is go to Google and then look at cell culture plates. So if you look at this, so you can see there are different um, sizes of these wells that you see. We'll just take this example. So this is one of the most common things you'll see. So there's a plate, and there are these six or whatever number of wells, and then you're filling them with some sort of a colored liquid, and it is inside these plates that you grow the cells, that the cells are grown. So whether you put that entire tissue within one cell, or whether you dissociate them, you are able to separate the cells from each other, and now whether they are as single cells inside this, either way. So this is what we talk about. These are culture plates: 6 well plate, 12 well plate. Yeah, this is a nice example. So 6 well, 12 wells, 24 wells. I think that's 48 wells, 96 and more. Okay. So whether or not I'm growing them, okay, and that colored thing, that's the medium. That's where your food and everything that it needs comes from. And we just talk about what all that is. Um, so when I put that in that culture plate, in that well, so now all five of them would not need everything, and they would not need the same thing. Maybe if one of them needs 10 things, the other needs 12 things, the other one needs 8 things, somebody needs 30 things, somebody needs 5 things. And if you draw like a Venn diagram for it, for what those 5 things need, so maybe I'll just draw this Venn diagram here. Is it working? Okay, so if I just draw, so let's say if that mass of tissue has 5 types of cells, okay, and now if I talk about their requirements, okay, we'll just draw like a Venn of requirements. So maybe if you look at something needs something, something, one of the cells need like a lot of things, it's very complicated. Something needs this one. Maybe one of them needs like this. Maybe one of them has something like very, very different. Okay. So now if you see, ideally, like in this figure, you don't see any factor which is common to everything. But even if there was something common, that is not enough, right? You do need uh like so many things. So now all five of them, well, can you provide all of these things? Maybe you can, right? So you have the option that you provide some of these things only, you provide all of these things. What a lot of people do is that they can separate out cells. So they'll grow this one in one. Like if you're able to separate cells, sort cells. So cell sorting is a technique. If you are able to sort the five different types of cells, then it becomes easier. Okay, you grow this cell with its requirements, you grow this cell with its requirements. All right. So it's complicated, right? If you want to, so if okay, there are two things here. One, how do you know what the cell needs? How did we find out that this cell type needs these 10 things to grow, right? Because you grew them in something basic, like let's say just sugar and salt, and it died. Like, okay, it needs something. You give it XYZ things, it gets better. You give it ABC, gets better, and so on. That's one hidden trial method, but again, that's not how it goes. You do know certain things that, okay, in the skin, we have these five different molecules floating around. Maybe the cell needs those five things. So that's one thing, that how do we know what the cell needs? Second thing is, what if the cell needs the other cell to grow? But so you need to figure out what this cell needs to grow, right? What if the second cell needs the third cell to grow, and so on, right? So finally, you need to grow all of them together, ideally, to get the best setup. So that's why tissue culture can be like tougher than cell culture, so as to say. Because if you're dealing with one type of cell at a time, maybe that's slightly simpler. If you want to deal with five different types of cells at the same time, there has to be some sort of a harmony, and you need to be able to support all of them together. And then what if something has a different growth rate, right? Something grows every day, something's dividing every day, something's dividing once in 30 days. What do you do? Do you start with more of something, less of something else? And so on. So these are just lots of things that need to be taken care of. So another, so once you've given a cell everything, it's growing, okay? Okay, once you have a tissue, you put that tissue in a medium, food, water, nutrients, some factors and everything. Cells, you see that there are, okay, cells which are growing. How do you know um, that the cells are behaving the way they should? Okay. So if you give me a minute, I'll just give you a figure. I'll just show you some images here. It did not strike me before that I should have shared that with you. In the meantime, if you have any questions, let me know. Otherwise, just give me like a minute. I should have something for you. Mm, Okay, never mind. I'll search for it and I'll probably then just show that to you in the next session. Okay, not important. Anyway, I'll just start sharing my screen again. Okay. Yeah, fine. So you're growing cells. You're looking at, okay, you are growing some types of cells. How do you know this is the particular cell type? How do you know it is a melanocyte, for example? Okay. So things that people consider when they're doing cell culture is, first of all, what do they need for growth? So food and all. Do they also need another cell? Do they need something which mimics that next cell? So in the lecture, the concept of ECM was introduced, extracellular matrix. So cells may not necessarily need the other cell, but they do have something around them which allows them to grow. So initially, what used to happen was, so the way a lot of cell culture was done is you grew one type of cell, it used to secrete that matrix. Then you used to remove the cells, leave the matrix in place, and then grow your main cell type on it. Okay, so generally fibroblasts, if you've heard again, skin cells, they are good at releasing that matrix. So fibroblasts are designed so that they grow lots of matrix, release a lot of matrix very quickly. Um, and then the cells are removed, the matrix stays, and then your actual cells, whatever's of interest, that is grown on them. Now, slowly, instead of growing that matrix right away, you get that matrix already pre-made. You can store it in -80 or -20, thaw it, put it on the wherever you want to grow the cells now, in whichever plate you want to grow them. Let the matrix solidify, settle, whatever, become in its final form, and then you grow the cells. So that's like a 30-minute job, maybe, rather than have you want to prepare a day in advance. Okay, tomorrow I want those cells, so today I need to prepare that matrix using fibroblasts. So anyway, some cells don't need those contacts. Okay. So yeah, so cell-cell contact, whether they need the actual cell or whether they just need the matrix from some cell. That's one thing. Second thing is, now whatever you're growing, so yeah, so okay, once you have, let's say you've established some culture in the lab, your cells are growing. Right now, they are exactly what is there that you got. Okay. Uh, right now it's the same thing, but then once it's dividing, it should still be what it was. Okay. Or if you're differentiating them, if you actually want, if you're starting with something like stem cells and if you're differentiating them, then that's a different story. But then what you have right now and what they make up tomorrow or day after once they divide, they should also stay the same. And then the $1 million question is, are the cells that you're growing right now, are they exactly how you say those cells are in the body? When you say you're growing melanocytes, okay, there could be some immature melanocytes, there could be some mature melanocytes in the body. Okay, immature ones, they're just coming up, and the mature ones are like, yes, we know how to do whatever. The ones you're growing in the lab, are they resembling the immature ones more? Are they resembling the mature ones more? Are they somewhere in the middle? Or do they have features of both plus also a third feature, right? Do they have a mix of melanocyte and maybe some other cell types composition? And the part B of that $1 million question is, how do you quantify that? How do you know if something is what you intended to be or if something is not what you intended to be? Okay. So a lot of things are there. One is you can talk about what they look like. So you directly take a tissue and then look at it directly under the microscope without growing it. And then you're growing that in the lab. And then you look at the cells. Are they looking the same phenotypically? Okay? Phenotype means what they look like. Second thing, you can test some function. Okay, if they are known to release that pigment, you can see if they're releasing the right amount of pigment. Are they releasing the right type of pigment? So pigment is nothing but some chemical structure. So there are techniques like, I don't know, mass spec that you can do. Okay, NMR, something like that. Those things can be done. So you see the pigment that's there in your skin actually, and then you can see the pigment that your cells in the lab are releasing. Another thing, you can look at the structures. So there's going to be something inside the cell that's going to be, where there's going to be pigment inside the cell, there are going to be structures inside the cell that are making those up. Okay. You can look at those subcellular structures. What else? You can look at, so I'm not sure if a lot of you may be familiar with this, but I'll just throw it out there and let me know if you want me to elaborate on it. There's something known as, so there's the central dogma is you have genes, so that's your DNA. From that, you get RNA, which is also called transcripts or transcriptome. And then those RNAs give you protein. So proteins mostly are the final things which are doing everything. They're the main workhorse inside the cells. So you can quantify those proteins, the proteins that you see in your cell, in your actual cell in the body, versus the cell that you see in the lab. Do they share the same kind of proteins? Are their composition the same? You can also look at their RNA composition. Okay, DNA you would assume is the same unless it's something like a mutation or something that you see in a cancer cell. DNA you would assume is the same. So that's also important. Over time, cells should not. So your body keeps these DNA mutations in check mostly, right? Your body is like, okay, if there's a chance that your DNA can get different, it's just going to kill that cell so that cancer doesn't happen. There are these checkpoints. There are ways in which cancer cells can evade those checkpoints. That's a discussion for some other time. But now, when you're growing them outside, that those checks can sometimes not exist. So those DNA mutations, they can go unchecked and they can now exist. And then what if you're growing a different type of cell altogether? You thought this was, coming from that patient we talked about. So you have that regenerative or no, personalized medicine thing, right? But actually, now there's a mutation in it, and it's no longer belonging to that patient. It's now a different cell, DNA like genotype altogether. Anyway, not going into too much details, but these are certain things that you would want to take care of when you're doing cell culture. These are the things that people have taken care of when they have done cell culture all these years. Protocols are made when they know that, okay, these cells need some contacts, some cells don't need some contacts. Cells need different types of contacts. They need, okay, you can talk about something that are called basement membranes, laminins, matrix, things like that. Different cells need different kinds of supports. So people have formulated that. If you Google different matrices, different ECM, you'll get a list that this is appropriate for the cell type and so on. People now know that, okay, what differences do they see in vitro and in vivo cells? By the way, just elaborating, because if you look at your assignment, there are a lot of questions which talk about where would you lie these processes into? Where would you categorize them? Is a process in vitro or whether it's in vivo? So simple example, like simple definition. If it's happening in the actual organism's body, it's in vivo. If it's happening outside the body, it is in vitro. That's it. Okay. We'll see some examples, not identical to the one in your assignment. We'll see some example today towards the end. All right. And then, of course, there are these checks. So whenever you're developing a new protocol, suppose today somebody is given a protocol how to grow neurons in cell culture. Great. Somebody objects, okay, we are growing neurons because they resemble your actual neurons in these 5 ways. But there is a sixth parameter in which it does not resemble. We are making a new protocol in which the neurons are now resembling actual neurons in all of these 6 parameters. 10 years later, somebody says, maybe not even 10 years later, 2 years later, somebody says, okay, but there's a seventh parameter that we tested, and now these neurons don't behave that way. Maybe the growth rate is different. Maybe their size is different. Okay, maybe they don't have one thing. Maybe they have some other different RNA or different protein that you normally don't see. Right. So now we are growing a different protocol. Anyway, so these are the checks that yeah. So these are the checks that enable better and better protocols so that you can better study your particular cell. Is there a question? No? Okay. All right. Let me know anytime, any question. You can unmute your mic or you can ask the question in the chat box. Okay. So we briefly talk about this, not in too much detail, also in the lectures, not even now. Cells, of course, they need some contact. Do the cells need another cell to grow? Do they just need some signals to grow? Uh, and does the cell just need some actual support to grow? Okay. Um, and so basically the thing is that the base of these plates, whether it's glass or plastic, depends, different tasks, different requirements. And why just these plates? Sometimes people also can grow cells on these glass slides or those cover slips. So if the cells, so talk about adherents and non-adherents. That's just fancy terms for sticking or non-sticking. So the cells here, they're sticking to something, right? It's not coming off. On the other hand, if you have a cut and there's blood flowing out when you touch it, of course that blood sticks to your other hand, right? So that is sticking. So that is, I mean, the other way. So it's loose otherwise, right? So it's sticking to wherever it can. It doesn't have, it's not stuck to its own self. Skin cells or most of your other cells, they're adherent. They need a surface, they need something to be sticking on. And glass or plastic may not give you that in the lab. So they need their own type of sticky material to grow on. That's what your matrix is. Yeah, so whether it's the matrix, whether it's other cell types, whether it needs some signals. In terms of those signals, you can like provide those signals outside. We now know a lot of things. There are things like growth factors, small molecules, signaling molecules, which tell those cells that okay, you need to behave this way, especially if you're working with stem cells. So I work with stem cells, so there's a slight chance I'll give you some of those examples. But let me know if you have more examples or if you want to talk about some other cell type and we can try it. So yeah, so there's lots of, so we know that, okay, these are the kinds of molecules which actually some cells secrete to tell this cell that, okay, you need to behave this way. So now you can externally provide those cells in that medium. So we talk about this medium, right? This, so these wells have certain medium inside them. So in that medium, provide those growth factors, some concentration, and the cells will sense that and they'll be like, okay, this is what we need to do. All right. So, yeah, so growth factors or signaling molecules and so on. And of course, nutrients. They need sugar, salts, and of course, they need O2 there. O2's there. All right. We'll talk about these protocols in more detail in the upcoming classes. But these are the basic things that cells need to survive. How far along are we? Just checking the time. Okay, 7 or 10. We have plenty of time. Okay. Cell cycle was also talked about. So there's, again, very briefly, we'll go into details of these things more and more in the upcoming classes. This was very preliminary just to introduce a lot of terms to you. So cell cycle is when one cell becomes two cells. As simple as that. Uh, sorry, I'm wrong. Cell cycle is not exactly one cell becoming two cells, but it includes the entire process. So cell cycle is where the cell prepares to become two cells, and then it eventually does. And then it's preparing whether it needs to do another round. So it's the entire setup. It's not just that division. That division process is what's called mitosis. Um, so yeah, so that entire thing is cell cycle. And there are cells which absolutely don't, which are actually not a part of that decision. So like they will not be dividing. So neurons, the final, like they're called terminally differentiated neurons. So they are neurons, they're absolute neurons, there's no stem cell feature in them. And they've differentiated and now they're not going to divide. They're not going to become two cells. They're too specialized in their function and in their design that it's very, very hard for them to become two cells. You've seen diagrams of neurons where they are all very, very fancy, right? So there's, again, just a small attempt, but so there's something like this nucleus, and then there's going to be a lot of these more and more complicated structures. There's dendrites and axons. Again, we'll talk about this when the need comes, right? And then you have got this generally one thicker outgrowth, which is called the axon, and then it's going to make contact with another such neuron, more neurons here and here and here. Okay, so this has very, very specialized things. It's not just this simple look. There are more structures here, and there are substructures like some things, some spines and all here. Lots of things. Okay. So the molecules you'll find in this part and the molecules you'll find in this part, they're all very, very different. So in short, neurons, these terminally differentiate neurons don't divide. Also, not all the times do your stem cells divide. The stem cells that are finally there in your adult body, they don't divide unless there's a need. There's not an always, like, for example, you say that the liver can regenerate. It can regenerate when it needs to, right? There's a basic homeostasis that, okay, you have new cells coming up from these stem cells, or you have new cells coming up from the pre-existing cells because they're dividing into. For new cells to come up, you don't always need stem cells. Pre-existing cells, your mature final cells can also divide. And in most cases, that is what is happening. But a lot of these times, these stem cells are out of the cell cycle, but they're temporarily out. They're not permanently out. Terminally differentiated neurons, as we understand them right now, they are permanently out. But there are trials going on, people are studying them, bring them back into the cell cycle. All right. So that's the basic difference between dividing and non-dividing cells. More terms, again, probably it's not the best heading here, but then things we've briefly touched upon this is differentiation. So stem cells becoming some lineage, more and more specialized lineages. Dedifferentiation is when you go back. So remember I told you this example of personalized medicine where you're getting cells from a patient and you're making brain cells, for example. And so that's partly what my PhD project is also on. So what's happening actually is you're not taking brain cells from the patient because that's invasive and you don't want to damage anything. So what you do is you take skin cells again. Skin cells are super helpful. So you take skin cells and you can de-differentiate them into stem cells. And now, so again, think it's the examples like I'm not giving any exact names and all here, but you're starting with stem cells. Yeah. And then let's say it divides into lineage 1. And there's, let's say, lineage 2. And then furthermore, you have lineage 1, right? It can become the second type. Maybe there's third type here. What else? Maybe there's one type here. And just one more type here, okay? Let's say this is how your, how differentiation is happening. So what we are doing, so when you're going, when you're going in this direction, this is differentiation. And when you're going in this direction, it's when it's de-differentiation. So just another term that I'm throwing at you is Yamanaka factors. So the scientist who discovered this reverse, one of the scientists was Yamanaka, and I don't remember the first name. So there are few things, again, some sort of factors, some molecules that you need to provide these cells, and then they can become stem cells. Again, not perfect stem cells, but that's the best we have at this moment. Again, how do you test if something is a stem cell, right? Can it become all of these 5 types? Can it become 2 types? Can it become 3 types? That's called the pluripotency, or the potential of that stem cell to become anything. Anyway, we'll talk about this again later. So differentiation is when it becomes specialized. De-differentiation is when you go back to that non-specialized state. De-adaptation is somewhere in the middle, so as to say. So adaptation, in layman term, when you use it in a non-molecular biology context, is when, for example, you're going to higher altitude, and there the concentration of oxygen and the pressure that's different. So you're getting adapted over time. You don't directly go to that 8,000-meter peak directly because your body is not used to that pressure. Or when you're diving, same thing goes there. You're not going there in a quick plunge. Slowly, you are acclimatizing your body to it. Similarly, you can have cell types which are adapted or which are trained to uh, respond to something somehow, and now you're taking that stimulus back. So now do they stay that way, or now are they de-adapted to that stimulus, like that? Again, this was not elaborated upon in these recorded lectures, so I will not elaborate on them any further. Another example that was talked about was about the ECM acting as a barcode, and what happens when you're talking about um, cells turning into cancerous cells. So you talk about our skin, for example, but skin, heart, brain, lung, more organs, um, they have different ECM requirements. For example, even within your skin, there's these different layers present. So you have um, the epidermis, dermis, and I think the hypodermis. These are three layers. Anyway, there are multiple layers even within each of them. So with different types of cells or different parts of cells. So for example, just imagine if this is a neuron. Neurons also, by the way, in your brain, brain also has different layers. I think 6 layers typically is what we talk about. So it's possible that one of the layers has more of these types and one layer has more of these types. That's also possible. So it's not that each layer has a particular type of cell, but it could have different parts of that cell because each part is doing a different function, right? These cells are not uniform in shape. So the two ends, they can do different tasks. Anyway, so each layer or each different tissue or organ, it can have a different ECM composition, which acts as its own identity. Barcode here simply just means the identity of those cells. But now when a cell becomes cancerous, it loses that requirement. And when we're talking about these adherent and non-adherent cells, so non-adherent cells are, one example is cancer cells, in addition to your blood cells. Uh, when you're culturing cancer cells in the lab, they don't necessarily need a base. Okay, we'll again talk about these in more detail in the upcoming lectures. So now they don't need that base, or the other option is they can work with any kind of base. So sort of, if you've heard about metastasis or metastasizing cancer, is the cancer actually originated, those cells, some cell in your skin originally turned cancerous, but then it broke off from there. That's not what normally happens. In a healthy person, your skin cells would not travel to the lung, but a cancer cell from the skin, which originated from the skin, it can travel to the lung and it can lodge itself there and then grow there. So that's metastasis, and that's something that people have not been able to find a good cure of. Then there's no control over where it can be. Yes, that's a story for again some other time. So ECM is quite important. That's one of the things which cancer cells are good at evading. Okay, just a couple more slides. So I've tried to just summarize the different cell types that were talked about in this lecture, in the first 5 lectures. Again, you don't necessarily need to remember these. A lot of this would come to you over time. Cardiomyocytes are the muscle cells in the heart. Cardiomyocytes, so cardio, heart, myo is muscle related. Cytes is the technical term for cells. Endothelial cells, these are generally water lining your blood vessels everywhere. These are endothelial cells. So skin has its different layers, the epidermis, dermis, hypodermis, epidermis. There are these 5 layers that were talked about in the lecture. I don't think you need to remember these names. It's good if you can. As far as I'm aware, you don't need to remember. So basal cells is one example of the stem cells that are there in the skin. Melanocytes are your pigment giving cells. Brain, I think we only talked about these terminally differentiated, that's the term, terminally differentiated neurons. But of course, there are lots of other types of cell. Again, so these are the cell types we were mentioned. Lots more in each of these tissues. Blood, you have erythrocytes or the RBCs, leukocytes or the WBCs, and macrophages. Food for thought, just try and see what is the difference between leukocytes and lymphocytes. If you already know that, great. Otherwise, just take a look. Leukocytes and lymphocytes. Then you have these pluripotent stem cells or just stem cells for simplicity's sake. HeLa cells, these are cervical cancer cells, and we did not talk a lot about this until now, but cells have a limit. Okay. So one of the reasons why everybody has a death eventually, one of the things is that cells have a limit. Okay, they cannot be growing indefinitely, and this is a preventive measure from there being overgrowth. So cancer is basically too much growth, right? That is what is different between cancer cells and normal cells, that they are not immortal. Cancer cells. So what is the benefit of something immortal? Is especially when you're dealing in the lab is I buy something. If I'm so cells, you can buy cells. Okay, there are these companies that you can buy cells from, and then you can keep it at a very cold temperature, so they just stay like perfect. They're frozen and they're intact. So let's say I have bought 10 tubes just for safety. Today I'm thawing one of them and I'm growing them, and then they can grow indefinitely. So I grow them, I continue growing them, and even like 2 years later, I can still grow them. They're not dying. They're not saying that, okay, done, bye. Um, that's what cancer cells and some cells uh, can do, right? So, which would be great in theory, right? Because I have an indefinite supply. I don't need to keep buying things. I always have something with me. But then, what's controlling that the cells are not changing something about them in those 2 years or in those 10 years, right? There could be one mutation. DNA, again, I don't think we're talking a lot about DNA in these lectures, but DNA is like, think of it as a big book, big novel, and we need to keep retyping that novel. Okay, every time your cells are dividing, you're typing another copy of that novel. Today you type one, okay, you're great at typing, you don't make mistakes, autocorrect is, doing its job. But then someday, somewhere you make a mistake and it's not detected as a mistake, right? For example, the word is HOT and you're typing as an HIT. So it's not detected as a mistake, right? Because that word still exists. The sentence doesn't make sense again, but the word still does, right? So you've evaded that autocorrection check. But now the cell is different. Now the book is different. It's doing something else than it should be. So that's what um, immortal cells, that's why immortal cells can be a problem. Okay, because those checks sort of vanish. Okay, we'll talk more about the HeLa cells. HeLa are the starting, the first name and the last name of the person, Henrietta Lacks, from whom these cells were derived. So HeLa. Primary cells, immortal cells, we'll talk about more in detail. But yeah, basic thing is primary cells, they have a limited lifespan. Immortal cells are theoretically immortal. And I think this is the final sort of slide. There were some processes in general that were also mentioned. So vitamin D, you have cholecalciferol, which is vitamin D in your skin, that goes to the liver, becomes something, that goes to the kidney, becomes something else. That helps the uptake of primarily calcium in the bone. That's why vitamin D is important. And then you need sun as the starting thing for this. So that's why they say that, okay, just taking vitamin D is not important. It's not sufficient. You need sun as well. Okay. Then, so telomerase was where we talk about So the ends of the chromosomes, they just keep cutting and chopping off. So you can cut the extreme end, you can cut something in the middle, more, but then you can't cut anything. So that's what controls the cells being from being mortal or immortal. If those things are getting regenerated again and again, then there's infinite cells for you. So that's what happens in aging. You have more and more of those chopped out. Telomerase enzymes do things. Again, not going into details at this point. Karyotyping is just looking at chromosomes. So looking at your DNA from a bird's eye view, just looking at it if they look okay overall. Not going into the exact details of the book. It's like you're looking at the contents of that novel. Okay, there are 10 chapters. These are the roughly the page numbers, the sizes of each chapter. Okay, it looks fine. Great. So that's still an important check. It's not always that you need to go into the details because you can already find some mistakes and just by looking at the contents. All right, so that's one. ROS generation was also discussed a little bit that we have O2, that we are using O2. So reactive oxygen species can be generated, which are not great, which can lead to DNA damage. That's why they're not great. Mutations, so on and so forth. Again, talked about briefly. Cell cycle, so that's the entire cell cycle, and then it sort of stack. So mitosis is when the cells are actually dividing, and then they are deciding whether or not they want to go through that cycle again. So G1, thinking and preparing. Synthesis is where almost everything is prepared in double, especially the DNA. That's the synthesis phase. And then G2 is more checks because you don't really want to be sure. Did we just make 2 copies? Did we not by mistake make 4 copies or 3 copies? It's like that. So that's like your gap phases, which are mostly checks. All right. I'll come to this thing in a while. So now we just quickly have very, very short questions. I don't want to keep you for too long. But yeah, here I would really want some of you to give answers. Again, these are all multiple choice questions, very similar to what you will see in your assignment if you already have it. But it'll be great if the people here can just tell me A, B, C, D, whichever you think is fine. Okay. All right. So easy peasy. Which of these is not a constituent of the animal cell? Yeah, so A, B, C, D, or 1, 2, 3, 4. Cell wall? Yes, 2. All right, great. So you know that plant cells have it, animal cells don't have cell wall. Again, this was not explicitly talked about in the course, if you think about it, but yeah. So cell wall is very specifically almost exclusively a feature of plant cells. Um, and some bacteria, I think the gram, yeah, bacteria also. Okay. Next up, in which phase of the cell cycle does the DNA replicate? We just talked about it. In the S phase, right? In synthesis phase. Okay. In which phase of the cell cycle, the cell either progresses towards DNA synthesis and then another division, or it exits the cell cycle to commit to differentiation? Any idea? Others can try. It's okay if you're wrong. No problem. Because I skipped this explicitly when we were just talking about the cell cycle. So does anybody know? So the exit that we're talking about here, that's the G0 phase. So it can be a permanent exit, like we see in the case of these neurons, or it can be a temporary exit. That, okay, right now I'm not dividing, but I can divide whenever I need to. No idea? Okay. So there is this phase. So if we draw it like a cycle, so it's M, and then it's G1, and then it's S, and then it's G2, and then it's back to M. So it is after M. So when the cell has just divided, it's then, so at So it can just make this decision of whether to go ahead and enter the cell cycle or exit the cell cycle. Okay. So the question is, in which phase? So it's during the G1 phase. Okay. So if I, yeah, so it's during G1, I should say. Or it's somewhere, something like this. Okay. Because I mean, I'm drawing these arrows here, but ideally, if you look at it, maybe I'll just remove this. So there's no gaps, right? So it's more like this pie chart where you have M and G1 and G2, right? So it's M, G1, oops. Sorry for the mess. And then this is this. And then so it's not, so it's during M, it's just M. But then it's during G1 that it can go up. So during the G1 phase is when uh, the cell can exit the cell cycle. All right. Dash implies that the specialized properties of the cell is lost irreversibly. You can still hear me, right? There's no response now. So yeah, it's okay if your answer is wrong. Just give it a go. You just need to type 1, 2, 3, 4. Come on, there are like 22 people except me in this lecture. One of you should give it a go. You can, and again, please, you can unmute yourselves if you want. So, so the specialized function is lost irreversibly. Uh, so here I, so 3 is a good try, but here we would talk about redifferentiation because the specialized properties are what the cell gets when it's differentiated. So when you're de-differentiating them, you're going back, and ideally, yes, they're not lost. Those properties are not lost. So this one would be de-differentiation. But now a similar question is, which process implies that the synthesis of specific products which are under some regulatory control and they can be reintroduced if the right conditions are provided, right? Now this one is de-differentiation. So yes, so question 4 and 5, they are confusing, but just see. So when we talk about irreversibly, we're talking about de-differentiation. So, and when I talk about this thing here, this de-differentiation here, so unless you provided again some specific way, specific things, normally they can't grow back. It's only recently that we have these properties that okay, they can, you can make anything out of anything. But typically you would assume that they're lost irreversibly, that they can't be reversed. It's de-differentiation. And if it's just about the right conditions, because there's some regulatory control, okay, some heat is what you're providing, or you're providing some, it's not specific factors, but just some signals, some sort of regulation, then that's about adaptation. Okay. Cardiac cells, what are cardiac cells mostly? What type of cell is your heart made of? Is it a bony structure? Is it a lot of neurons? Is it a lot of muscles? Is it a lot of epidermal cells? It's a lot of muscle cells, right? Yes. So cardiac cells, most of the cells are cardiomyocytes, muscle cells. Which of these gases, along with its buffer, is used to maintain the pH? So again, this was talked about in the lecture. Yes. So carbon dioxide. So the idea is that carbon dioxide and water is going to make carbonic acid. H2CO3, is it? Right? Hmm. So yes, this is H2O. and your carbon dioxide, that's going to become H2CO3. So that's carbonic acid. Okay, so that's why this is the right answer. So yeah, so CO2 is an important part, and we will talk about how CO2 is given to cells. Um, CO2 is explicitly given to cells when they're being cultured. Okay. Which of these is a non-adherent cell type? Osteoblast. So these names were not exactly talked about, but osteoblast has something to do with bone. Hepatocytes, I think it's liver. Erythrocytes is blood, and neurons are, well, neurons, brain. So which of these is a non-adherent cell type? So erythrocytes. HeLa cell was derived from, we just talked about it. So this is a cervical cancer line, right? So that's true here. And okay, so this is, can you just classify these into in vitro and in vivo and in vitro processes? So in vivo is inside the organism, in vitro is outside the organism in a lab environment. Okay, so can you just identify the in vivo processes in this? It'll be great if one of you can unmute yourselves. It'll be just quicker rather than typing. Just saying. Please feel free to unmute yourselves. I'm not asking you to switch on your cameras or anything. Just a quick answer would be great. Because you have like, I think 8 or 9 or 10 questions in uh, the assignment. So just to give you an idea. So yeah, so wound healing is in vivo. Electrophoresis is in vitro. So electrophoresis is in the lab when you're looking at DNA flowing through a gel. Drug screening on culture cells is also in vitro. That's right, because now you have cells which are outside the body. So if you're screening, this is drug, wrong spelling. And immune response. So we talked about immune response coming from antibodies. So while the antibodies may have been made outside in vitro, but the immune response that's generating is being generated inside the body. Right. So that's in vivo. So thanks, Anushka. Those are the right answers. And thank you for being interactive. All right. So I'll just label these. So this is in vivo and this is in vivo. And so the other two are in vitro. All right. Okay. All right then. So that is all I had for you for this week. Again, if any of you joined us after our initial discussion, so the session that we have had today was recorded. So that we are currently recording it. So I will upload this recording on YouTube. So Pallavi UQIDAR, that's my YouTube channel, and then I'll just make a playlist for this course. I'll share the link again. And then this PPT today, I will put on this Google Drive. Again, let me share the link with you. But also, these links will be available to you through, sorry, where is this one? So if you go to this problem solving session recording page, there's going to be this click here option, which is going to open this page. Now it's empty right now because we've not had any session until now. But then I'm just going to fill this form for you. You'll have the YouTube link and the presentation folder link here. Join this discussion forum if you already haven't. There are going to be questions. Maybe people ask similar questions. So you can ask questions if we already don't solve them in these classes. The deadline for assignment 1 for this week is in 2 days. So don't forget to submit the assignment. And there are generally between 10 and 20 questions in each assignment. Yeah, it's especially important if you want to have a certificate for this course, because in addition to your final exam, marks from these assignments and participation from these assignments is necessary. There's one more live session that's going to be happening tomorrow. Most of the things that would be talked about in that would be similar to this. It's just going to be taken by another PhD student. So Mondays or Tuesdays, whichever one works for you, you can go ahead with that one. And I will see you all again in the next Monday session. So that's going to be the 1st of September. Same time, same YouTube, same Zoom link. And so yeah, so that's the outline for these Monday sessions. So then for the next week, in addition to discussing the concepts from week 2 and talking about some practice questions from that week, we'll also talk about the current week's assignment because the solutions would have been released by then. And then so that's another thing we'll add on from the next week onwards. Yeah, so that's about it. Again, if you have any questions, please feel free to unmute yourselves now or later. And yeah, I mean, I'll still be here for another couple of minutes. You can anytime, any question, you can put it in that Q&A forum. And that's all. And thank you everybody for joining in. Thank you all for being such a sport. And I'll see maybe some of you or all of you next week. All right. Thank you. You can leave the class if it's all done. Otherwise, I'll end the meeting in a couple of minutes. Thank you.